What do cells need to live in the environment that surrounds a typical cell? The cell membrane and cell wall are two structures that surround a typical cell. The cell membrane occurs in both plant and animal cells, while the cell wall is found only in plant cells. The cell membrane exists to protect the inner contents of the cell. The cell membrane also helps to hold its shape and regulate cell growth by balancing endocytosis and exocytosis. The cell wall provides structural support for plants, fungi, and algae, whereas animal cells lack a cell wall. Cells are typically surrounded by fluid, carrying essential components including, but not limited to, carbohydrates such as glucose, fatty acids, enzymes, hormones such as testosterone, neurotransmitters, amino acids, and ions. The environment will change depending on the location of the cell. The cells will require different resources for mechanisms depending on the cell's location. The surrounding environment of each individual cell can vary, especially for those needing different concentrations of the available components. It becomes clear that the cell would need resources such as nutrients, molecules, or elements from the surrounding environment. Bacterial cells have cilia or flagella and can move through the environment due to these structures. What features form the basic design of a simple cell and will address a cell's basic needs? For many cells, structure is directly correlated to function. It is very important for the simple cell to be able to at least store genetic information, although it would be most efficient if the cell was also able to self-replicate that genetic information, a process known as DNA replication. It would be important for a basic cell to be able to address energy needs, as well as cell division and replication, as well as waste removal. Cellular glycolysis and respiration are required processes, as ATP is produced via these processes. ATP is necessary for all cellular functions. The genetic information must store instructions to make or retrieve nucleic acids necessary to form the genetic material, be it DNA or RNA. Transcription to interpret proteins such as mRNA and tRNA are also important features. Another important function of a cell would be waste removal, as accumulation of waste can lead to cell death or disease. Basic cells have needs that would require the function of organelles such as the nucleus, the endoplasmic reticulum, ribosomes, Golgi apparatus, peroxisomes, lysosomes, cell membrane, and the mitochondria. What biosynthetic processes are needed to fulfill a cell's basic needs? Cell respiration is a necessary process, as production of ATP provides energy for the cell by utilizing oxygen and glucose with a byproduct of carbon dioxide. Exocytosis is the excretion of biomolecules out of a cell, as accumulation can lead to cell death or disease. Endocytosis is how a cell absorbs nutrients, is also necessary for the basic survival of a cell, as many different biomolecules are required for many cell processes. A basic need of the cell itself may not include mitosis or meiosis if only survival is necessary. Cell division and replication may be necessary for the survival of a species or group of cells, but not the individual itself as an artificial cell. If our only goal as scientists is to create a single-celled organism that can survive on its own, cell division and replication may not be necessary. What parts are needed to build the minimum machinery of a cell? First, we begin with a nucleus. The nucleus holds all the DNA and coding information required to build necessary machinery for the cell. It holds the genetic information, in the form of chromosomes, and prepares the cell for execution of the instructions within the information held. Without a nucleus, we are unable to reproduce, repair, or create the machinery required for function. Next, we have the ribosomes, which is an organelle located on the rough endoplasmic reticulum and also in the cytoplasm, which synthesizes proteins with the help of mRNA. Then we have the plasma membrane, which is a semi-permeable lipid bilayer that acts as a protective function for the cell. Proteins and other machinery may exist on the membrane to add channels and other functions to allow for certain molecules or ions to be transported across the membrane. This enables the cell to create an internal environment separate from the exterior. We have the endoplasmic reticulum, which is a structure extending from the nucleus. It synthesizes proteins and lipids. The endoplasmic reticulum also aids in catalytic reactions with lipids. We have the Golgi apparatus, which is considered a subsection of the endoplasmic reticulum. This is a protein transport mechanism that packages and marks proteins for transport in and around and away from the cell. We have peroxisomes, which aid in creation of oxygen and water for the cell through breakdown of fatty acids. We have lysosomes, which are digestive organelles that break down macromolecules into their smaller components. And we have the mitochondria, which are considered the powerhouse of the cell and home to ATP production, which is important in every single cellular process that requires energy. How do we build this machinery, and how do we tell whether or not the synthetic cell works? To be able to create a synthetic cell, the cell must include the critical features, organelles, and biosynthetic processes that we mentioned earlier. The mitochondria may be a feature of the cell that might need to be replaced. This may be done with lipid replacement therapy. LRT has been studied and has shown that lipids have the ability to repair and improve mitochondrial damage. In theory, we could study this further to see if lipids have the ability to fully replace or replicate mitochondria, and this may help us create a synthetic cell. In the assignment, our goal is to make our artificial cell glow green. Essentially, we want our synthetic cell to exhibit bioluminescence. One way our cell can exhibit this trait is if it had the principal pigment luciferin, an enzyme such as luciferase. Luciferin undergoes an enzyme-catalyzed oxidation, and the resulting excited state emits light upon decaying to its ground state. We need the pigment luciferin and the genes required to produce the enzyme luciferase. Success may be measured by determining if the cell's processes are fully functioning and it is able to produce a bioluminescent glow.